And thanks for coming to the last talk of today. My name is Fereydoun Hormozdiari, and I'm from UC Davis. And I usually work on structure variation. That's on discovery of structure variation and copy number variation. But this talk is a little bit different. It's about what is the contribution of structure variation on genome structure. So first, let us define what structure variation is. And tomorrow, we're going to have a couple of talks talking all about structure variations. But in short, structure variations is defined as large and medium-sized genomic rearrangements. These are genomic rearrangements that change genome when the length of the arrangement is usually bigger than 50 base pair. And they come in different flavors. They include, for instance, things like deletion, when a segment of genome is deleted, or insertions, or tandem duplications, different types of tandem duplications, inversions, translocations, and so on. These are what we call structure variations. It's known that structure variations can have significant impact on disease and evolution and about this sort of functional effects. When structure variation, the question is that how do structure variation contribute to different phenotypes, like diseases that we mentioned, like autism, cancer, and so on? What is the mechanisms that they contribute? Majority of the studies that have been published on contribution of structural variation on diseases or other phenotypes look at the contribution using coding regions. In other words, how a structural variation affects the coding region and results into a phenotype. And that's easier to interpret because we know a deletion of a gene or duplication of a gene has a direct effect on its expression and thus on the phenotype. But what about the coding regions? How does the structure variation which does not affect the coding region can contribute to disease? And we know there are quite a few of them which do that. So with advent of high-C technology, we are able to study how a genome folds and how different regions of genomes interact with each other. And that gives us an ability to study how different regions like enhancer and promoters regulate expressions. I will go over how a high-C technology works and what is the output that it produces. First, assume that we have the genome which is folded in this three-dimensional space. What they do is that they add some chemicals that results in different regions that are close to each other are cross-linked. In other words, these regions that were in three-dimensional space physically close with each other are linked. Then by adding restriction enzymes, this genome is shattered such that the regions that are cross-linked with each other remain cross-linked, remain linked together. And then these segments are given to sequencer. First, they are ligated and then given to sequencer such that the paired-end sequences that are produced by sequencer, one end comes from one of these links, the other end comes from the other link. So now you have this paired-end data that sequencer has produced, which means that these two ends are physically close to each other, not necessarily in the genomic distance, but in the physical three-dimensional distance. These two ends are close to each other. Now, if you consider these two ends and you map it back to a reference genome, let's say one end of the paired end maps to location I, and the other end maps to location J, it means that in three dimensions, there is at least a little bit of support that this location I and J are close to each other. So you can show this in a matrix fashion. Let's say X and Y in this matrix are the genomic position. And position I and J is the number of the reads that support location I and location J being close with each other. The higher this number means we have more support that position I and position J are close in three-dimensional space and potentially are interacting. So this is how a real high-C matrix will look. This is a chromosome 10 by chromosome 10. Each location gives you the number of the reads that we have seen in this experiment. The first thing that you can notice is these boxes, these squares, are very intense red. This means that these regions are highly interacting with each other, or they're very close with each other. So there was a name that was given to them. They were called topological associated domains, or TADs. And they're quite interesting. As I said, there are regions in the genome that we know are highly interacting with each other. Again, these are highly interacting with each other, but they usually don't interact in between them. So this region was studied more. So here's what we mean. Let's say this is the genome. This folded in three-dimensional space. This is one TAD. This is the other TAD. As you can see, this region is interacting with each other. They're very close. This region interacting with each other is very close. But between them, we have very few interactions, as you can see by not having that many paired end reads crossing them. All right? So this is what TAD is and how we will think about it. It was very interesting that they observed that these boundaries that they call TAD boundaries or TAD insulators, they're highly enriched in CTCF binding sites. There are locations that CTCF binding sites that bind in this location in genome, they tend to be enriched in, the TAT boundaries tend to be rich in these elements. So it's believed these elements have a major contributing factor in making sure that these TATs are created. Okay. Any questions till here? Please feel free to interrupt me if you have any questions, anything is not clear. Because this is important and this have a major effect on how we define our problem. CTCF is a protein that binds. It can go and bind there, and there's a mechanism that makes sure that the location that they are bind are not able to interact with each other. So it's a protein that goes and binds there.
So your function is to make sure
Yeah, it has many functions. It has several different functions. One of its functions is in the 3D structure is makes sure, and in fact, there are a lot of papers that have been published in the last 2 years that exactly try to explain how it works. There are some very nice animations, but this protein works with another one called cohesion, and it makes sure that this structure is created. So it works with another one. But yeah, in this context, its main job is to make sure that that these TATs are created. For instance, if you delete this CTCF, if you don't have CTCF, you will not see these TATs so nicely. It will all just mix up. Yes? Oh, it varies. It's usually between around 100, 200 kilobase per to 1 megabase per. That's approximately, but it can vary significantly from different TATs. But that's the approximate thing, between 100 kilobase per to around 1 megabase per. Okay? So going back to the question of how non-coding structure variation can cause disorders, one idea is that let's think about how genes are regulated. When we have enhancer or promoters, these enhancers fold when the genome folds, and they interact with the gene, and they result in the expression of this gene. That's the general model that you can think about how an enhancer can regulate a gene. But then, if you think about it, if you have these TATs, and we have these enhancers, and we have these genes, we said that these boundaries make sure that different regions of one TAT are not interacting with another TAT. In other words, if you have an enhancer in this TAT, it will not interact with this gene, and thus it will not regulate expression of that gene. Each enhancer inside a TAT potentially is only regulating the genes that are inside the same TAT. But if you have structure variations which disrupt this organization, potentially that can cause into dysregulation of the gene and will cause a potential disease. For instance, if you have two TATs, you have gene 1, gene 2, and you have enhancer. This enhancer in this TAT is controlling this gene, and it will not have an effect on this gene. But if you have a structure variation here, although it's not necessarily affecting the two genes, but it can change the regulation and thus change what we expect. So there's one very nice, I think the first paper that shows very nice studies of this was this Lopez et al. In Cell, but there are some very nice Nature Review Genetic papers that show this result, especially the one that was published very recently. So if you're interested in this topic, this is a nice paper to look. So they have three real examples of cases that structure variation exactly cause these sort of, using this phenomena cause disorder. This is the case number 1 I'm showing. We have the boundary, these are the genes, and this is the boundary. And if we have a duplication, for instance, this region is duplicated, they call it the creation of a new TAT, because if you assume this is duplicated, now you have a new TAT here, and that resulted into disorder. You might have a deletion if you have this boundary which is deleted. Now you had these enhancers which were controlling this gene. Now they might go and control this additional gene. This was another example using a deletion. Or you might have an inversion, which you had segments. This region was not interacting with this one. Now you have an inversion. Now they do. So these are three different structural variations, which all three are disrupting TAT structure and resulting in 3 different diseases. Most of them are usually development disorders, because the phenotype usually tends to be quite strong. Yes?
And there are actual results that those genes become regulated by those
Yes,
enhancers?
yes. You should look at these papers. They do a lot of wet lab studies in mouse and stuff to show exactly that after these events happen, this enhancer starts to regulate it. You see the expression of the gene goes up. And they show the interaction. So all of this is done in those papers. Yes, for sure. If you're interested, this paper and the Cell paper exactly show this stuff. This one. Okay, so going back to our problem, we wanted to study more how specifically deletion can change the TAT structure, and we call that TAT fusion because we have two TATs, similar to what we had gene fusion. If we have a deletion of this boundary between X and Y, this enhancer affecting this gene, we call this creation of a new TAT as TAT fusion. So we have the high C in the genome H, which is unmutated. We have the position of the deletion between X and Y, which is potentially deleting a TAT boundary, creates a new TAT, and we call that a TAT fusion. And in this scenario, this enhancer is affecting this gene and potentially causes the disorder. But we want to quantify this. We want to be able to quantify these types of structuration, how much they're contributing to this phenotype. So we define what we call a TAT fusion score, and we define TAT fusion score as the expected changes in number of interactions, pairwise interactions, as a result of a deletion. So a TAT fusion score of a deletion between X and Y, we define it as changes in the expected number of interaction in the genome before the deletion minus the expected number of interactions in the genome after the deletion. So this is the value that we want to calculate, and this is the TAT fusion score that we are defining. Is this clear, the definition? Good. Okay. So our goal is to measure this expected value of changes in the interaction due to a particular deletion. So our input is the high C matrix H, which is the unmutated genome, which is the matrix, the high C matrix before the deletion happened, is the coordinates of the deletion between X and Y, which is called delta X and Y. And the output that we want to give is the score of the deletion. In other words, if these two TATs are fused or not as a result of this deletion, we want to be able to give a score for that. And the framework that we are suggesting has two main steps. We say at the step one, we try to predict how does the high C matrix change as a result of deletion. In other words, if the high C matrix before was H, we try to predict the new high C matrix G as a result of this deletion. In other words, we want to have a model that we have, if we have the high C matrix and we have the deletion, what is the new high C matrix after deletion? And then, given the two high C matrix H and G, we want to estimate the number of changes in the interaction as a result of this deletion. In other words, what is the difference between number of interactions given these two high C matrices? So the framework is quite simple. We just want to first predict how does the high C matrix change, and given the two high C matrices, how much of an interaction change we are seeing. So for the step one of the method, which is about predicting the changes in high C as a result of deletion, we have to model the interactions between every pairwise high C value observed in the matrix. And the way that we're going to do it is using, because there is no real model at the moment exists, we're going to use the published material of what we believe are contributing to these values. For every I and J, the value of their interactions is known first and foremost is relevant to the sequence content of I and J and their normalization that's going to happen. So we're going to have a parameter for each being I and J that we're going to try to learn. Also, it's been shown that the number that you observe in the high C matrix is very dependent into the distance between I and J. The farther the I and J it is, the lower connections you're going to see between them. It's something that it's been observed, and it makes sense. The farther I and J is, the less likely that they will interact. And it was shown that the value drops in a power law fashion based on the length provided. So we have a parameter beta, which tells us based on how much far we go, how much of a drop that it will. That's by the beta is a negative value. And also, because we have these boundaries between TAT boundaries, we have to model that. So we define a parameter Ri, which is the resistance. It tells us for every I and J, what is the resistance or TAT boundary between them. In other words, if we have more resistance, we expect to see more drop in their interactions if we go in between them. So these are the parameters that we have, and what we want to learn is, given the high C matrix, we want to learn the alphas, we want to learn the betas, we want to know the distances, and we want to learn the resistance. And the way that we want to learn that is we want to optimize the observed value difference from what we're going to get using the model that we have. So we can write this as a simple minimization function, and we want to learn these parameters. And the distance between I and J, we're going to assume it's a Euclidean distance, so it has to obey triangle inequality, the distance between I and J. And if you can see, this is a very, very hard problem to optimize because you have a logarithmic term here, you have the absolute value, so this is not linear. But what we can do is that we can just simplify this problem by changing this Dij, the distance between I and j. The easiest, we use two different approaches. The easiest thing you can do is, of course, assume the Dij is the genomic distance between I and j. If you assume the Dij that we're gonna plug into this is just the genomic distance, that's very trivial. We know between every I and j what is the distance. Or we can try to approximate their physical distance given the high C matrix. So there was a paper published in Nature Method a couple of years, maybe a year ago, which used shortest path between, calculating shortest path between every I and j to calculate the physical distance between every I and j given the matrix. So these are the two different Dijs that we can plug into this model. As a matter of fact, we tried both of them and they work reasonably well. The shortest distance approach worked slightly better. So the results that I will present are based on this approach. But using the genomic distance worked reasonably well. So now, if we assume in this model we have the Dijs, in other words, the Dijs are given for every I and j, it's not a parameter, then the model becomes quite easy. It becomes a linear programming. So we can now optimize this as a linear programming and calculate all other parameters, alpha, beta, and r, which minimizes this difference between what the model gives versus what was observed. Any questions about this optimization formulation? Is there a question? No? Okay. Okay. So if this is the model, if this is the parameters that we learned given the high C matrix, now the question is, if we have the deletion between xi and j, can we recalculate this matrix? It should be quite easy, right? Because everything remains the same. The only difference is now the distance is different. The distance is now going from Dij will be going from Di to x and from y to j because this is missing. If you were using only the genomic distance, it would be very easy to calculate. If you're using physical distance, we said this Dij is smaller than equal summation of this guy plus this guy. In other words, we took the upper one. And then you get rid of all the resistance here because they don't exist anymore to have a resistance. In other words, if there's hat boundary here, it's gone. So from this part of the formulation, everything from x to y is gone, and everything else remains. So now we can recalculate the high C matrix using the model that we have after the deletion. Any questions? Okay. So if we have assuming that we have the high C matrix, then we have solved our step one problem. We had a high C matrix, we had the deletion, we calculate the new high C matrix after deletion using this approach. Then the question is, what is the expected changes in interactions given the first high C matrix minus given the first high C matrix and given the second high C matrix. And we can model this using the calculating the probability of interacting for every I and j given the high C matrix. And for calculating these probabilities, we use the known set of loops to calculate what is the probability of I and j interacting based on the database of set of loops that we have and the high C values that we have. So this part becomes easy. We can just learn this function using the available databases of loops and plug it into these functions that we have. So given the two matrices, now we can calculate what is the expected changes in number of interactions given the two high C matrix. Okay, so this is the method that we have developed. Any questions about this part before I go into results? About the step one of calculating the high C matrix and then calculating the score? Okay.
Can you just repeat what f is?
Oh, f is the function. F is the function that we learn for probability of interaction between I and j given the high C value for I and j. In other words, if in the high C matrix you observe a value of 10, what is the probability of I and j interacting if the value is 10? What is the probability of I and j interacting if the value is 20? And the way that we built this function is based on looking at, because we have a database of interactions that we know, and then we look at for those interactions, what is the what are the values in high C matrix, and we built the distribution. So having a good distribution of loops or interactions is critical. We assume we have a database that we can use. The better set of loops that we have, the better set of interactions we have, this f will be better, and the method works definitely better. Okay, so the input data that we use for high C, we use the Rao et al. 2005 data for NA1278. This is one of the highest quality high C data available. And for deletion sets, we use 3 different sets of deletions. We use the 1,000 genome set of deletions as a control, in other words, the normal population set of deletions. We use the deletions from TCGA cancer deletions. We use the deletions from deletions that are fixed in grade A lineage. In other words, the ones that during evolution have been fixed, and we expect those fixed deletions to have the least amount of TAT fusion because they're fixed. So going to results, the first thing that we tested was that how does our model work for predicting the high C? In other words, how does the step 1 work? And because we really don't have that many high C data available from different samples with deletions, it's hard to test this, but we did this. So we came and assumed for every location, if we assume randomly pick a location and consider all the interactions on the left of all the high C values on the left of this location and all the high C values on the right as the input or known data for training, and use that for predicting the interactions in between them. So we didn't consider a prediction in between going through this line for our training model, but we used that as a testing and wanted to see how good our model works. So the green is the result that we get for our model for predictions, and the blue is that if you only use the known drop based on the distance for predicting the interactions. As you can see, our model does way better than naive approach. This is the PCC result, and this is the L1 distance. Our model does way better for predicting interactions than using the naive method of only considering the distance between I and J and trying to predict the high C value. We also looked at the one cancer cell line, K562, that we have high C matrix and we have a set of somatic deletions. We only consider the deletions that the length is above 5 kilobase pair and are validated. So there are 5 of them that we have. And these are the values for our model, the PC for all model, the distance based, and the length based is the naive approach. As you can see, again here, our approach does better than naive approach. And these are the PCC that you get for 2 of those deletions. Yes? Correlation. It's a Pearson correlation coefficient. Yeah. Between what was observed and what is predicted. And I would emphasize this, when you look at the simulation, our PCCs looks way better, like looks around 0.9. Here, it looks around 0.5 or 0.6 in real data. And the reason is that this is cancer cell line. There are too many genomic rearrangements happening in cancer to be able to really predict amazingly. There are just too many different variations are happening. Here is a very clean simulation, so it's, of course, this will be the best we could do. This is, I would say, one of the worst results we could get because it's comparing a non-cancer cell line versus cancer cell line. So this was about comparing our first step, which was seeing how good our method or model works for predicting high C values. But there are a lot of methods that have been developed for calling TAT boundaries. Why we couldn't just get those methods, predict the boundaries, and see if the deletions hit a boundary or not? So this is the four tools that have been developed for predicting TAT boundaries. And we ran these TAT boundary callers on the 1 kg deletions. And if they deleted a TAT boundary, we said that's a TAT fusion based on their method. As you can see, the amount of overlap between these 4 tools is very, very low. The amount one that only agrees with all 4 is only 4, versus a large portion of them is only predicted by one of them. So or all tools have a large false scurry, or they're missing a lot. And that's kind of has been shown in the past couple of years that TAT callers tend not to agree with each other that much. So that's why we are using a method such as we developed for calculating a score for fusion. Okay. I will skip this. This is the result I would like to show. We then came down and looked at the scores that we get, TAT fusion scores that we get for all the deletions, which is the y-axis, and compared against the deletions that different tools called as TAT fusion deleted the boundary. So if none of the tools predicted a deletion, there's around 7,000 deletions that no TAT caller predicted that it's deleted boundary. So this is here. If 1 tool predicted, if 2 tools predicted, 3 or 4. As you can see, if there are more number of tools that are predicted a deletion to delete a TAT boundary, our method was successful at giving it a higher score. So this correlation shows that our method is adding value in being able to predict the TAT fusion. For instance, if you believe that more tools are doing a better job in predicting TAT fusion, our method is also giving a higher score. But I would like to emphasize, this is nice, this agreement is nice, which shows our method is truly finding something interesting, is working correctly. But also, if you see, there is a large portion of deletion, a large portion that no method is predicting as deleting boundary, and that's expected. Only a very small fraction of deletions should be TAT fusion. However, there are some that show that our method has given a high score. If we looked at some of them, and here this is 2 examples of them. This is the deletion, this is the high C, and just by looking at it, it seems this deletion really is deleting a boundary. So we believe our method is doing a good job of giving a high score, although there is no TAT color has predicted a TAT boundary there. And then I look at the CTCF binding sites. Both of these have very strong CTCF binding sites using ENCODE data. So this is another evidence that potentially this is really a boundary and this deletion can cause fusion. But now going to some biological result. The first biological results that we checked was we wanted to see if TAT fusions really are bad. This means that in evolution, they should be getting rid of. In other words, if you look at the deletions that are fixed in great ape or deletions in 1 kg, we should see less of them than what's randomly expected. So we looked at the deletions that are fixed in great ape, and we randomly permutated this deletion. The same size set of deletions were permuted in the genome, and we ran our method to calculate the score for them. And we did that many, many times. So this is the y-axis is number of the deletions that had a TAT fusion score above a particular threshold, and these are the thresholds. So the blue boxes are the number that we get using random permutation, and red is the real number using the real data. As you can see, for the real data, for the most of the high TAT fusion score, for all the different scores that we compared, as long as it's bigger than some threshold, after that it will not hold. The number of the TAT fusions that we see is less in real data than what you would expect from random. This means in great ape, in fixed deletions, the ones that have very high TAT fusion score, they're being getting rid of. The same principle holds for 1 kg data. These are normal patients, normal people, so you don't expect to see too many TAT fusion in them. Similar principle holds. There is a less number of TAT fusion or deletions with high TAT fusion score than what's expected by random. And interestingly, so we believe this shows that there is a TAT fusions are under negative selection, and 2 papers one was published in GR just now using gibbon genome, and the other one that was in bioRxiv from Katie Pollard's group, which they compared again the great ape data, showed very similar result, that TAT fusions or deletions that change TAT structure are in their negative selection. We looked at it in a different way too. We looked at the allele frequency, because in the 1 kg data for all the deletions we have the allele frequency. We wanted to see how does the allele frequency compare against the score. So the x-axis is the TAT fusion score, and y-axis is allele frequency. As you can see, if the deletions have higher TAT fusion score, they tend to have lower allele frequency. This is another indication that if you have a very bad deletion with a very high TAT fusion score, it doesn't tend to become fixed. So this is also an indication that these deletions are potentially are selected against. But also, if you remember, the reason that I motivated this problem was a set of deletions that people were reported that these deletions is resulting into disorder because of disrupting TAT structure. So we looked at the literature and we found around 7 or 8 that were published in different papers who were claiming such a phenomenon, and we ran our method to give them score. So the blue is the result from the 1 kg deletions, and the reds are different published deletions that were reported to cause disruption in TAT structure and cause disorder. As you can see, all of them are getting way higher score than what you get from the 1 kg data set. Finally, we were interested to zoom into these deletions. So we picked 2 of these deletions that were very well studied. And these are the high C data. These are the deletions. These blue boxes show the deletions. You can see this deletion exactly is fusing these 2, or this deletion is fusing these guys. So it's clearly causing TAT fusion. But we said, what if we break this region into small deletions of 20 kilobase pair? So in other words, we break this into 20 kilobase pair and run our method for every 20 kilobase pair and see what score we get. So this is the deletions. Every 20 kilobase is a deletion, and the y-axis is a TAT fusion score. You can see the bump. It's it's almost 0 for all the deletions till you reach around the boundary. When you reach around the boundary, suddenly the TAT fusion score becomes really, really high, which very nicely matches the CTCF binding sites that have been reported in ENCODE data. The same holds for here. You see the TAT fusion score gives the highest score here. Here it has a really high jump, and here, and it agrees with, again, with CTCF binding sites. So we did that for all 8 deletions. We looked at the deletions that, the 20 kilobase per deletions that remove CTCF binding sites versus the ones that didn't remove CTCF binding sites. And we can clearly see that the ones that remove CTCF binding sites are getting higher score than the ones that did not remove CTCF binding sites. Okay. Finally, so we talked about, we looked at the great ape data. We looked at the 1 kg data, and we compared the TAT fusion score for deletions between them. And that 1 kg deletions had higher TAT fusion score than fixed deletions in great ape. And that's something we expected because we believe this has gone through, these are old because they're fixed in great ape versus some things that you see in 1 kg data. But then we looked at the TCGA deletions that are in cancer patients. And then we can see that they have the highest TAT fusion score because there's no selection acting on them or very, very little. And you can see they have a very high, and also it might be even beneficial to have a deletion to increase the production of cells and things like this. And all these comparisons were, of course, significant. And the differences especially were nice when you compare the TCGA versus 1 kg deletions. We also did the exact same comparison for the TCGA versus 1 kg, and we broke it based on the size. We tried to look at the size of the deletion to make sure that we don't have effect. And you can see always the TCGA is more than 1 kg. For most of them, it's significant. Some of them, it didn't reach significance because there wasn't enough deletions. But okay. So in conclusion, I defined what we call TAT fusion score. And I explained the method that we have developed for calculating TAT fusion scores, which is based on first calculating a new Hi-C matrix after deletion, and then comparing the 2 Hi-C matrix to predict the number of the changes in number of interactions. And we showed evidence that the TAT fusions we believe, are under negative selection. And in disorders that have deletions that cause TAT disruption, we are giving high score to them. Some of the improvements that we are working on is that we really would like to have a better job in making prediction for changes in Hi-C. And one way to do it is to consider other biological markers like CTCF binding site, histone modification markers, to use this in our model to improve it. At the moment, we are just using the Hi-C data. None of the other things are being considered. And then the biology becomes very interesting to look at in great ape because we have all these fixed deletions. We know some of them we are claiming that have a TAT fusion score, but we also have the expression data. Does this deletion really change the expression as we believe it should? So compare this, which we haven't. Finally, this approach at the moment, the method that a model I presented was only for deletion, but you can extend that for other types of structural variation, for instance, for inversion, duplication, translocation. And if you think about it, you can see how the resistance and alphas, if you have a deletion or inversion or duplication, you can also extend the model to work for them too. And yeah, that's my final slide. And I would like to acknowledge the postdoc in the lab who did most of the work, who is not here, Glenn. And that's it. I would be happy to take any questions.
Question, Fereydoun?
Yes.
What effect do you think somatic mosaicism in those deletions or the structural variants would have on all of your predictions about the effect on CAS? I know that's really important.
Sure. So except the TCGA data, all of the other were germline, so they were not somatic. Only TCGA, and I think that's a great point. So let's say you only 10% of your cells do have that deletion. So only those 10% will be affected negatively. So one reason that maybe we are not seeing this effect here because maybe it's not, the allele frequency is not enough to have a significant effect. But also that might be the reason why we didn't do that much of a good job in predicting Hi-C in the K562 because it was a somatic, right? Maybe the deletion is only affecting 10% of the genome. But so how would I model this is a very hard question, but definitely its effect is significant in this analysis. Yeah.
So you showed that your TAD fusion or TAD fusion score corresponded well to the CTCF binding sites. How frequently did that happen the other way around, where your score is low and the binding site is there or detected?
So in the very high TAD fusion score, at least the top 100 or 200, almost all of them have CTCF binding sites. But this doesn't mean that all the CTCF binding sites are TAD fusion. Only around 15% of CTCF binding sites are believed to be boundaries. So there's a lot of CTCF binding, the majority of CTCF binding sites are not necessarily bounding. So I think based on our analysis, around 50% to 20% max for really boundaries. But the ones that got very high score, all of them had CTCF binding sites. So one idea is to use CTCF binding sites for improve the method, but we haven't done that. Yes. Sure. So to clarify something, we are not trying to predict the TAT boundaries. We are trying to predict if the deletion results in TAT fusion. So we are not necessarily, so we don't care to say, oh, definitely there was a TAT boundary there. TAT caller's job is that. But to be able to compare, we said if you predict a TAT boundary and deletion matches there, you're assuming that you're predicting a TAT fusion. Versus we don't make the TAT caller prediction. We just say, oh, this is a deletion. We believe it causes fusion based on a model. So that's where the difference is. Or the genomic information is not helpful in predicting the TAT boundary. Just saying, oh, here is a deletion. What do you think? Does it cause a fusion by deleting a TAT boundary, or based on our method just predict a fusion? Yeah, exactly. So I use them as a way, because there isn't really a database except those deletions to say, oh, these are the TAT fusions. So we use this to say, okay, look at these high calls that they said definitely is a boundary deleted, and we can also predict that as a TAT fusion. But there's a lot of disagreement between them, and we show that potentially we do a better job than do. And there is a reason for that, because TATs, although I showed it very beautiful pictures of these triangles, in reality, it's very hierarchical. So there are TATs, and there are super TATs, and they're nested. So a TAT caller that only tries to pick boundaries will have a hard job. But for us, if we are only going to give a score that things getting disrupted, we don't care if it's nested or not nested. If it's fused, we say it's fused. Yes. Yes. Not necessarily. It's the genomic distance. If it's in three-dimensional, they interact or not. The distance is correlated, but it's not the only thing. So in the three-dimensional, if they interact, then it will get regulated or not. The distance is important. The DIGA, the distance is important part of the formulation. Although we got a better result, which we use the genomic distance versus the physical distance instead of genomic distance. That gives slightly better results.
Okay, my question is, you said that for all these types of variations, it's similar to deletions, the effect of deletions, and you can usually modify this to some mode, but they don't exactly get them closer together, but some of them will get them.
Sure. So but you can predict how the distance is changing, and also you can predict how the resistance are changing. Let's say you're duplicating something and you had the resistance here. The same resistance you can assume came here. So you can change the distance based on that change. Yes. Gian. Oh, sorry. Mm-hmm. Resistance, yeah. Yeah. They reflect very much, they reflect TAT boundaries, but not sometimes. I mean, we looked at the TAT boundaries that were predicted by a lot of tools. A lot of parts agree, but some parts they don't. And as a matter of fact, what we realized was that sometimes you see the spreading of the score. For instance, you don't see one bin with a high score, but you see 4 or 5 bins connected will have a little bit score going up. So there's a correlation, but it's not exactly. It's highly correlated with TAT boundary, of course, but it's not that if you run TAT colors versus 4 or 5, there's a lot of them that they don't agree to. Yes.
Well, you showed the results where you have high TAT fusion score, but none of the other methods have detected a signal there. What do you think is the reason there? Because you showed in the later slide that these sites has
CTCF. Yeah.
Boundary, but since you also explained only half of the CTCS how do you know you are not able?
Sure. So you're right. This might be false, but first of all, this is only not this one. This is based on, these two examples based on visualization that this is potentially two tasks, because currently there is no way to really know unless you go and do the CRISPR to know that two is correct. But based on this indication that there is CTCF and based on visualization, we believe this is more probably this results into TAT fusion. But yeah, till you don't experimentally validate it, you won't be sure here.
Why do you think the other methods use that?
This is my guess right now based on this visualization inspection right now. Based on this visual inspection that this potentially seems to be two TADs and there's CTCF binding. This is a TAD boundary that they don't have it. And the other things that they're extreme disagreement between them. That we know they are missing a lot.
Thank you very much.
Yeah, thank you.